Luca was a cell, the last common ancestor, the last unknown common ancestor. I always forget the U. So this was the ancestor that made all of our ancestors, right? All of our other cells. Every living thing that exists. But what came before it? So Luca has DNA, and it has organelles. Well, actually, no, it was a prokaryote, so no organelles. It was basically just DNA with a cell membrane. So what came before it? Well, maybe it was just DNA. Maybe DNA gave rise to Luca. So let's explore that idea. What is DNA? DNA is essentially just information. DNA is made of tiny little parts called nucleotides. Those nucleotides we'll go with that. Those nucleotides connect together with phosphates to each other. And so if I were to draw those in here, they would be all over the place. It's called the phosphate backbone. And they're on both sides. This is really tedious. So it connects, each one of these is called a nucleotide. And then you see these little sticky things here, right? So let's look at that. On each of those nucleotides, we have what's called nitrogen bases. And those nitrogen bases can be A for adenine, C for cytosine, G for guanine, and T for thymine. The rule goes that, so these basically connect to each other in a DNA molecule. And so the rule goes, if there's an A on one side, it'll connect to a T, and a C will always connect to a G, and vice versa. And so that is how this is formed. So that right there is information, and it can be passed on. So this DNA can be passed on to your offspring. And so possibly this was the first self-replicating molecule. But here's the thing. Here's how DNA copies itself. Only portions of DNA make something. At specific locations we call loci. We have what are called genes. A specific sequence of A's, C's, T's, and G's are genes. These genes, they're information. Those genes can be read and turned into proteins. Proteins are very big, and they have a specific shape that does a specific thing. And so sometimes there's a specific protein called a polymerase, because it makes polymers, and that can actually make more DNA. But the problem is, so this is DNA making itself, which means it's a good candidate for the first self-replicating molecule. However, we have a chicken or an egg problem. So this is the problem, right? Let me erase all this. DNA can make more DNA, but it needs a protein to do that. And so the protein also needs the DNA. Protein cannot exist without the gene on DNA. And then the DNA can't exist without the protein. So it's a chicken or the egg thing. What came first? Well, most likely, it was neither. Most likely, what came first was DNA's close cousin, RNA. RNA, the first thing you'll notice is that RNA is only single-stranded. The next thing is, if we look at each of its little parts, it does have A, and it does have C, and it does have G on the little nitrogen base right there, but it has U instead of T. DNA had A, T, C, and G. So what's different here is the U and the T. So that's one difference. Another difference is that it's single-stranded. And one really cool thing about this is that RNA can actually fold on itself. You see all of these bonds in here for DNA, they're tied up. But RNA can actually fold on itself, with those A's going to U's and those C's going to G's. Furthermore, it can connect to other RNA molecules. Excuse me. This can create a lot of complexity. And so you can think of it like a balloon that a clown twists. RNA can be twisted into intricate shapes that do intricate things. When they do so, they are often called enzymes. So most of the time, your DNA can make a protein enzyme, but RNA itself can become an enzyme when it folds like this. And so an enzyme is a molecule that is reusable, meaning that it doesn't break down upon use. And it physically alters a substrate. So for example, this RNA molecule here might grab methane. And it might add methane to something. And so it's useful. So this would be the substrate. And this would be the enzyme. And so it grabs a substrate and it uses it to catalyze a reaction. So catalyze means to speed up a reaction and make it possible. Catalyze is to reduce the activation energy. And we've talked about activation energy before, and there'll be more on that in a second. But basically, during any reaction where there is time and energy, a certain amount of energy is needed before the reaction can occur. Right? So, actually, I drew that a little bit wrong. There's always reactants and there's products. And then a certain amount of energy is needed, that's the hump, for it to occur. But when you catalyze a reaction, you reduce that amount of energy and it happens faster. The hump is reduced. I hope this all is a reminder from biology. So anyway, RNA is the likely candidate. It reduces activation energy. Another cool thing is that this folding process right here, where RNA can fold on itself due to its self-binding, is spontaneous. The only part that isn't spontaneous is when the nucleotides of RNA connect. They connect just like DNA with these little blue things here. It connects with phosphates. And that process requires energy, and we just don't know how that happened reliably enough for life to occur. So that is one mystery. So anyway, when you make a RNA enzyme, we just call it a ribozyme. It just makes sense. All right, there you go.